So in my last lecture, I was talking about that a genome is all of the DNA in an organism, and a chromosome is just sections of that that then contain the genes to code for directions. So I want to talk a little bit more about chromosomes. So as I said in my last lecture, not all of the organisms on the planet cut their chromosomes up the same way. So we could all just have one big chromosome, but that makes it very difficult to organize and find locations for, like, locations of genes and so forth in a cell when you're looking for directions. So all of the organisms actually cut them up into these different size chromosomes. And corn, for example, has 10 unique chromosomes. And even though it has only 10 unique chromosomes, and fruit flies, those little fruit flies that we started off the semester with, have only 4, their actual genomes aren't that different in size. So it actually doesn't make any difference how big your genome is. And we'll actually look at here in a little bit about, for example, the difference between a fruit fly genome, so the total size of the genome between, say, fruit flies and humans. The size doesn't matter as much as how much information is actually there that can be used. All right. And so the number of chromosomes that you cut your information into doesn't really do anything other than help you as far as the way it's organized. For example, dogs and chickens have 39 different chromosomes, while goldfish have 47. And if you think about it, these organisms aren't, it's not like one of them is super complicated compared to the other. Right? You would probably say, goldfish might not be as complicated, say, as a dog, but it has nothing to do with how complicated the organism is or how big their genome is on how many chromosomes they've evolved to put that information into. And then, so my question is, how many chromosomes do humans have? Now, this is looking at the unique chromosomes. So how many unique chromosomes do we have? So in my previous lecture, I talked about that we have a total number of chromosomes, but then we have a unique number of chromosomes. So how many unique chromosomes do humans have? And it's 23. So we have 23 chromosomes. So we've taken our genome and cut it into those 23 sections versus, goldfish is 47 or dogs 39 or fruit flies 4. And the thing is, all of the individuals on this screen actually do the same thing that we do. They inherit one copy of each chromosome from each parent. So that means, for example, dogs and chickens actually have a total of 78 chromosomes, while humans have a total of 46. Okay? So the number of chromosomes can vary widely between organisms. Now, I also want to talk a little bit about genes. So genes, talked about previously, are those sections of the DNA that code for a characteristic or a trait, and alleles are just different versions of a gene that code for the same feature. All right. So, just so that we're all on the same page, traits are any single characteristic or feature of an organism. So in this example, we're looking at flower color. Put me up here. We're looking at 3 different flower colors. The gene is for making the color for the petals. So that's the gene. The allele are the different colors. Okay? So the gene itself is the same in the sense that the gene is making the coloring for the petals, but the allele is going to make the petal color orange in this one, or yellow in this one, or purple in this one. And I don't know if this will help make a little more sense, but if you think about genes like recipes for cookies, right? So we can have a basic cookie recipe, say like a sugar cookie recipe. That cookie is still going to be a cookie even if we add chocolate chips. Yeah, it'll be a chocolate chip cookie, but it's still the same sugar cookie, just with chocolate chips added. And that's the idea here with alleles, right? Is that an allele is the same basic recipe, just with slight changes to it. And so, you know, with the orange petals, something small has been changed to make that color orange versus yellow. Right? So a slight change in the pigment or an addition to it to make that pigment a little bit more orange. So it's reflecting more, say, red than the yellow one. That's what's going on here. Okay. So let's look at an example of this in humans. And not just in humans, but other animals as well use melanin. So melanin is a blank responsible for skin pigment. So this is the thing that's made, right? So we're looking for the product. So we're talking about, again, genomes, the genome, the chromosomes, and the genes. So what is it that genes make? Genes make blank. That's going to get you the first blank. So melanin is a blank responsible for skin pigment. So this is part of a trait or a structure. People who have albinism, so people who are what we would consider albino, or animals that we would consider albino, carry a mutant blank that does not make melanin. So this is related to the directions. All right. And in this case, the best answer is C. Since melanin is a product that's making skin pigment, it's got to be a protein. All right. Since proteins are the main thing that are being, made to make up our structure and function. So it's got to be a protein. And then people who have albinism, so they can't make this, means that they carry a mutant allele, a mutant set of directions, a mutant gene that does not make melanin. So now let's take a little bit more of a closer look at the genome versus the genes in organisms. So I told you we were going to take a look at our genome versus things like fruit flies. So here we are. So here's the fruit fly genome. And these numbers are in millions of base pairs. So you can see down here, millions of base pairs. So that's millions of A to T sets, C to G sets. And you can see that the fruit fly genome is not nearly as big as, say, a human genome. So 180 million base pairs versus 3,400 million base pairs. And like I mentioned before, fruit flies, they cut that into 4 chromosomes versus our 23. But if you look at something like an onion has 18,000 base pairs. And that's huge compared to humans. And I know that onions don't cut their chromosomes into an insanely large number. Think about like corn. Corn only has 10, and it's a little more along the lines of an onion. Salamanders, on the other hand, you can see have 84,000 base pairs, and an amoeba is a single-celled organism, right? And they have 600,000 base pairs in their genome, right? So think about that. A single-cell organism has an insanely large number of base pairs in their genome compared to something like a fruit fly or a human. So your genome size and your chromosome number has nothing to do with how complex the organism is, right? It just all it's telling you is how much genetic information you have inside of your cell. Okay, so for example, just to give you an idea of what this number means, that means that it has, almost 200 times as much DNA in it as something like a salamander. A salamander has 25 times as much DNA as we do. So that, is a huge difference, but it doesn't make a difference as far as how complex any of us are. So let's take a little bit closer look at what is making up that DNA. And the main thing that we look at with DNA are those genes. Because remember, the genes are the directions. If you take a look at this chart, which organism has the largest amount of DNA that's actually coding for proteins, that's actually made up of genes? And it's the E. coli. E. coli has 90% of its DNA made up of genes. Versus us, humans only have about 2% of all of our DNA that is actually coding for our proteins. So that means, I'm sure you figured it out, 98% of your DNA in every cell is not genes. It's not coding, is what we call, it's non-coding, meaning it's not really directions on how to make stuff. I mean, a fruit fly uses almost 20% of its DNA as genes. And a fruit fly has a smaller genome than we do. So what's going on? Well, we're not 100% sure exactly why this has happened, but we do know that prokaryotes like E. coli and other bacteria have somewhere up to 90% or more of their DNA as genes. So they usually have 10% or less of their DNA in this non-coding part. So let's take a little bit of a look at what's going on here. So like I said, bacteria and viruses, both, genes make up 90% or more of the DNA. What is the rest of it? Well, eukaryotes have what we call introns. Now, bacteria and viruses have them too, but they're very small comparatively. Introns are these sections of DNA that don't code for proteins. And about 75%, as you can see here, of that is in between the genes. It would be kind of like if you had written a paper and you had a paragraph of that paper that, was actually the text of your paper, and then you just randomly typed on your keyboard for the next page or two, and then wrote the next paragraph of your paper. That's what our genes have, or our genetic information has done. So we have these sections where our genes are at, where there's information written there. And then there are these big spaces where there's non-coding information, we call these introns, before you get to the next section of the genes where there's information to be read again. Now, you can see too, there are sections of introns inside of the genes. And so those are sections of information that are just kind of almost gibberish in between the information in the genes. So let's take a look at what those introns are. Like I said, bacteria and viruses have very few of these, but eukaryotes, we're stuffed full of these. So these are non-coding regions of DNA, so they don't have information on how to code for any genetic thing like making proteins or anything. There's a couple different things that can happen. They often take the form of short or long repeated sections. So they could just be sections that just say A, like a record that's gotten stuck, you know, where the needle's gotten stuck and it's just jumping. And like it said, it can just repeat thousands and thousands and thousands and thousands of times. And that doesn't have any information in it. Just like if it was a needle on that record jumping, right? You only got a little bit of information, but not enough to like sing the whole song. The other thing that we find are things that look like genes, but aren't. So it might be a section of a gene, so a gene fragment, where maybe part of it got copied and then the rest of it got destroyed for whatever reason. We also find duplicate versions of genes in these introns. So, you know, extra copies that aren't being used. They've been turned off and just have been left to be dormant. And then we also have pseudogenes. Pseudo for fake. And so these pseudogenes are fake genes. What they are is that when we read the genetic code for them, they look like they should code for a protein, but there's just enough of them that is broken, right? There's just enough in the code that's wrong that it doesn't actually make a functional protein. Basically, it's a gene that has been mutated too far to actually make anything functional. We've also got gene fragments and pseudogenes that have come from viruses that have infected us in our past, as far as like our past ancestors that we've gotten passed down. We'll talk when we get to microbes about how viruses infect us, but they infect us sometimes generally by giving us genetic information, and that genetic information can get integrated into our cells. All right, we're going to finish this section off with a question. So which of the species here has the least amount of junk DNA? So which one has the smallest amount of those introns?